Okay, so next, this video, I want to kind of put things in a way where people can understand and kind of a simplistic way of thinking about these different terms and having a quick reference for understanding and not having it be confusing, right? So I'm gonna just go ahead and say and tell you guys how I kind of think about this and sort it through in my mind, even being one who was very saturated in this entirely, right? So for like leopard geckos, bearded dragons, iguanas, other fish, but anyway. So first here, I want to talk about what's the difference between like an actual gene and polygenics, right? So polygenics, the best way to think about it is like if you're looking at an animal or a fish, let's just use an animal, it's easier to reference. So like the ears, or on a fish, you could be like the fins, and you want to elaborate those or work on slowly selecting, a different degree of whatever visual you were working towards. Or, like if it was red, you were like, oh, I want to increase the reds in it, and I want to work on, maybe slowly introducing other colors and line breeding it so that it's like red, black, and whatever else, right? So that here is going to be technically a polygenic thing, and that should be considered line breeding at that point if you're going to be using related blood working together, like the systems here that I showed you. This is you creating a true bloodline, right? Selective breeding you can do for enhancing polygenic traits, yes, but there is some differences as we go down here, right? But again, I'm talking about easy ways for you guys to understand, think about, and relate to. So selective breeding, the best way to think about it is, let's come back over here to this model. Let's assume that this was a non-related group, non-related group, non-related group, no shared blood or ancestry, right? So, okay, you would breed these two together. Offspring, offspring, offspring. And each pairing you would be selecting out for the visual traits that you liked, but in fact, you're just crossing a lot of non-related blood, getting the different results and selecting out that way and moving forward with those and then recreating the same process, right? So that is selective breeding, because you're not actually creating a line. This can be a tool to getting to the point where you want to do this, or you could just start with this. So many different styles, so many different reasons, and the complexities of what you would be doing too are just drastic. So, and like I said, I will be explaining the differences of all this. It will all make sense to literally anyone watching these as to what I'm talking about. So now, what would be considered a gene? So a gene is going to be something that can go against polygenic, right? That would surface and like force it to change visual, right? And that could be eyes, body appearance, number of different things, okay? So, or color pattern just completely changes up. That would be a gene. It goes against polygenics, but polygenics can work towards modifying this visually of how it would express and vice versa. We'll get into that later. That's another video. So a gene. Good examples here for you guys to think about would be albinism strains, different morphs, so like how they would visually appear. Like I said, genes that actually physically change with one or two copies of the gene, the visual of the animal. Eye mutations, things that would be considered, like I said, going against, so like blue eyes, brown eyes, those would be considered in this for humans, right? So let's say for like fish, so you're producing yellow eyes. Okay, great. So now you have an eye mutation. So examples of this for leopard geckos would be, for example here, so you can see the difference. There's a lot of different ones. Leucism too does that as well. There's other, depending on which animals you're looking at, there's like a whole massive list. So you guys get the point. Leopard geckos actually has each one of these here that is its own eye gene mutation. So you got NDVE, Eclipse, Marble Eye, and Blue Eye. These are all very, very distinctive. If you breed visuals to the other one, and the other ones are obviously not hit for those. Again, this is think of eye traits like albinism traits. You do not want specimens who are actually carrying multiples of these genes, because you will have a hell of a time sorting that all back out. It's totally possible to do. That's true. Yes, it's doable. I mean, everyone in the history of these systems being constructed, including myself, had to be part of the learning curve for this all developing here. But just the same. So you want to keep these isolated, and that's the importance of all this system that I was showing you. And in a previous video, I told you if you surface a new gene, how to test it and why, and the different processes for you to be able to determine these things here, right? So they would help you decide what are you working with, okay? So now that we understand what a gene is, then you have dominant. Technically there's codominant too, but we're just going to keep it simple so that everyone can understand, because codominant's not really good. So anyway, dominant would be complete with one copy. So like, let's just use, because I mean, I have a really strong background, and the genetics of these cichlids is nowhere near as advanced, like I said, as a lot of the other animals that, or even it's not as advanced as any of the animals in my past that I've been in. So cichlids is still in the very beginnings, believe it or not, of its true genetic journey as far as where we can bring this. So again, all these systems that I'm showing you here is how you move whole industries forward into the next one. People haven't even begun doing really some of this, yes, very little. So anyways though, we'll get into all of this stuff, like I said, later. But think about dominant as if you took one visual and you paired it to another one, and you magically had copies of this trait for the adult there, but the other adult didn't have that trait at all. So again, I already showed you guys how to test for recessives too, so in the previous video, so we're just going to assume that you watched that. So a recessive gene is going to be one that's not complete with one copy, meaning both parents must express the trait or carry it. Recessive. In order to show the gene, has to be both parents carry it. One parent can't have it, the other one not, and you see it. So can't be complete with one copy is the best way to look at recessive. Dominant, one parent can pass this. Again, polygenically, yes, if you had two that were similar, or whatever it was, you know, both parents would obviously pass polygenic traits onto their offspring as well. That's different though. These, again, can force entire mutations to it, right? These are different than this. So I just wanted to kind of show you guys that, so you guys could see the differences between true genes versus polygenics, the styles of breeding that would be, you know, what would be utilized in enhancing polygenics, and then the difference between dominant genes and recessive genes, right? So you could almost say that the polygenics is traits, not genes. I'll hold this here just so you guys can actually see it, screenshot it. But again, genes changes the visual hardcore in some very distinctive way and would very easily be able to be tested and proven to be dominant, codominant, or recessive. But the main two that you're going to be looking for is dominant and recessive. If you can prove that, then you can go on to later, and that's not applicable with these fish at all. And I don't know, it would probably be so many years later, but it doesn't matter. So anyways, polygenics, traits you would be line breeding for or selective breeding for to enhance. Dominant gene, one copy of the gene will recreate itself on the next offspring. Now, there are ones in certain species where if you paired two copies of a dominant gene, it would create a super form from compounding on itself, creating a new visual. Now, here's an example of this. So, Max Snow, on its own, white, black, can be muddied with color due to the influences of polygenics, right? But it does still ghost out the background. So, because the gene, Max Snow, is changing it. But the eyes will be normal. if it had eclipse in it, it would have snake eyes or some kind of varying degree of alteration on the black section of its eye, right? On one or both eyes. So now let's say that you bred 2 Max Snows together that did not have eclipse in it, right? You would end up with Super Snows, which would then be a completely new visual on the entire animal. Taking bigger splotches, and polygenics played a pretty decent role in how muddy the white background looked, and suddenly you would end up with a bunch of spatter black all over it, and the whites would be even stronger, and polygenics would play a significantly less role in the visual of what you were seeing. Now, here's the other thing that it does. It makes the eyes solid black. So, how would that then, just so you guys can see the correlation on why all of this is important, how would you then know if this animal here that did not carry eclipse at all actually carried the eclipse gene? You wouldn't know. So, then what you have to do is you have to take that Super Snow and you have to breed it to an eclipse visual. If you pop out eclipses, you know this is actually eclipse. If you do not, you know it's not. Do you see why this system here is so vital to understand? And no matter what life throws you during your breeding career, it's really important to be able to understand what is going to happen and what to do when you get there, right? And so, like I said, I mean, I was literally living this, breeding during this, and helping to even be a part of the community, discovering all of this kinds of things in several industries. So we didn't have the luxury of someone coming through and showing, like I'm giving you guys all these tools now. We didn't have it. We had to create it. Do you know how hard that was? How many real lawsuits there was? How many? There was no structure. People were isolating up genes and owning them, right? Because before you've released it, let's say that you have an official date online, right? And you're claiming it publicly, that's yours, and it's widely accepted as you're the one that surfaced that gene. What do you then do? That's your property, right? That's your kind of like copyright type deal. Right, so this is a mess. You have to have a structure. You gotta have a structure. You gotta know what to do when things get to a certain place. You have to understand these concepts. I can't stress enough how much the cichlid industry is seriously just like hindered to all hell. They are not advanced. Very simplistic. It is very early. Like I said, believe it or not, it is very early in the life cycle of the industry for cichlids. We are not messing with these enough. But in order to do all this, you have to start having a model that you follow and break down. And again, different genetics, I got news for you. It doesn't really matter if you're looking at leopard geckos, betas, cichlids, goldfish. If you are well versed in genetics, you will be able to translate over concepts like I was talking about lemon frost. I think I brought that up for you guys. Lemon frost actually being a gene that was realistically similar, the same, as another gene in beta. Creating like dragons, the white, high white background, and the problems with it creating diamond eye and such. Right, which is now also in what? Mollies. You see people breeding for the eye pigmentation trait, which is a mutation. And they're getting diamond eye. It's building up guanine crystals, iridophore, and it's causing the eyes to basically glaze over and crystallize. Same thing that was happening in leopard geckos. Same thing that's happening in betas. So I hope you guys can see here, you gotta know this stuff no matter what. And I am truly sorry that this industry hasn't had this kind of development, and I assure you I can understand the frustrations, because like I said, I was actually having to go through all of this stuff during the development of multiple industries, different types of animals during the entire exploration of all of this. And I learned through the hard way. So let's just say that. But I do hope, and I like to look at it as a positive though, I hope that you guys can see these things, you can absorb these things, and you can use this to now go out there and kick some butt. And I want to see you kick some butt, because I'm going to be kicking some butt. So let's get places. And instead of being nasty to each other and competing, trying to shove the other one down, why don't we be strong together and actually try and get each other to be able to have achievements to advance this industry forward? That's all I got to say about that. We should be compelling everything forward, not keeping it standstill or relying on some other country or who knows what. We should push forward strong to get where we want to go. And a lot of people in this industry specifically don't even know where it could go, because no one shows you this. They always try and, but I got news for you too. They also don't understand this. So that's besides the point really. But the point is, the development's not there, and I'm sorry that it's not, but it is now, and we can push forward strong together. I'm going to keep making these video series for you, and I'm going to be going over so many topics, it's not even funny, to where when you're done watching this, you can actually just go create what you want to do. And when you run into a roadblock, you're going to know exactly what to do. You're going to have everything I didn't have, and everyone else in the industries that I'm talking about. Leopard geckos, green iguanas, snakes, bearded dragons, African fat-tailed geckos. I was there, all of these things. We had to do all of this from scratch. Concepts, breeders from around the world had different concepts and stuff, but there was nothing else. So sink or swim was how it was. So I hope you guys having the tools will help this industry actually shove forward strong and quick. And I look forward to if some of you guys actually do surface out new genes, and you can always, as I said in every video now, if you do, you can reach out to me and I will help you. I will show you the steps. I don't want money. I don't want nothing else. I will show you what to do so we can move forward, not stand still. So I hope you guys are all having a good day. This is going to be the last video for me. I'm sweating my butt off. It is extremely hot out here. Literally, like, it's 100. Last time I checked, I think it was the 102. It's just heat, 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 heat. But anyway, so I hope you guys enjoyed this video, and I will be making some, yet again, every time I'm going to be doing more, and we're going to keep getting more advanced as we go. And the concepts that I'm going to keep teaching you too as we go, they are my own, my own long proven skills that got me where I was when I was in each of these industries, which in leopard geckos was just insane. Like I said, global genetic study. I was the only United States representative. And I was proposed to, not vice versa. They had already watched all the provings that I did publicly, all across everything, for everyone that was, you know, like, considered to be the biggest bigwigs and the foundation of leopard geckos themselves, proving these things to be faulty as our understandings and correcting as to what it was. So let's kick some butt together.